This is a video lecture accompaniment to the review T. Follicular Helper Cell Differentiation, Function, and Roles in Disease, published in Immunity. This video lecture is an uncommon format, and so an introduction is in order. This video lecture will cover the same overall content as the written review, but is not a verbatim recitation of that review. The same figures will be used, but I will not be directly citing individual papers. All references are in the written review. A table of contents is provided. Each of these sections is covered in the written review. I'll also be covering them here, but in case you have specific interest in just one of these topics, you can easily jump to the topic of interest based on the timestamps. TFH cells have become very interesting cells in immunology over the past 5 years. While they were first reported really in 2000 and 2001, widespread acceptance and interest in these cells was really only occurring after the identification of BCL6 as a lineage-defining transcription factor of TFH cells in 2009, and with it the demonstration that TFH cells are essential for most aspects of B-cell help and critical B-cell responses. And subsequently, in the past 5 years, there's been a huge explosion in activity in this field, resulting in many findings and consensus on a number of topics. Most generally, TFH cells are required for germinal centers and therefore the bulk of B-cell memory, both B-cell memory, memory B-cells and long-lived plasma cells. And as they are required for germinal centers, they're essential for the somatic hypermutation and affinity maturation of B-cells that occurs there, and therefore they're essential for most high affinity antibody responses if those responses are T-cell dependent. And in multiple experimental contexts, TFH cells have been found to be limiting for this process. And so not only are they essential, they are frequently limiting, and therefore from a biomedical perspective, that makes TFH cells of particular interest, because if one is interested in vaccine development and generating high-quality antibody responses, there's evidence that T-cell help, TFH cells, is limiting for that, and therefore enhancing TFH cell frequencies or function is a logical line of investigation to pursue. In contrast, or on the flip side, from an autoimmune disease or allergy perspective, there's also evidence that TFH cells can be limiting for those processes, and therefore from a therapeutic standpoint, focusing on attempting to block TFH cell numbers or function is a logical course of action as well. And indeed, there's now evidence that TFH cells have important roles in a wide range of diseases and biological processes, a few of which are highlighted here. They're important for protective immunity against infectious diseases by way of facilitating protective antibody responses. They are important for regulation of intestinal microbiota, again, by regulating the amount and quality of the antibody responses in the intestine. They're important for numerous autoimmune diseases, both in animal models and there's evidence in humans. They most likely have important roles for allergies, as they are important for the production of IgE. And most surprisingly, there were 2 studies published last year that found in humans, in 2 different types of human cancer, there was a strong correlation, as strong as any other biomarker. There was a strong correlation between the abundance of TFH cells at the tumor site and the long-term survival of cancer patients. And so this, along with other data, indicates that TFH cells may have substantially broader roles in immunology and immunity than facilitating antibody responses per se. And we'll come back to this when we directly deal with diseases later on. But this is to provide context of why TFH cells are interesting. And so because of all of this, there's been great interest in better understanding TFH cell differentiation and function and molecular biology and cellular biology. And so we'll start with TFH cell differentiation. The most important thing to understand about TFH differentiation is that it is a multi-stage, multifactorial process. There is no single event or signal that generates TFH cells. There are three well-defined stages of TFH differentiation. First, TFH differentiation generally initiates at initial dendritic cell priming of T cells, wherein the T cell receives signals from the dendritic cell or other APC that can result in differentiation towards an early TFH phenotype during the first couple days post-priming, or alternatively receive different signals from the dendritic cell or other APC and subsequently differentiate into a Th1, Th17, Th17, or peripheral Treg cell, all of which are generally programmed to be primed in a lymph node, we're showing a lymph node here, or spleen, but then leave the lymphoid organ and go to the site of infection or inflammation, right? That's the job of those cells, is to respond to an immunological insult and traffic to the site of inflammation or infection. Whereas in contrast, the job of a TFH cell is to stay in that lymphoid organ and provide help to B cells. And so there's this early dendritic cell dependent phase and resulting in early TFH cells that interact with B cells either at the TB border or within the follicle. And this is occurring before the development of germinal centers. And then there's a third stage of differentiation wherein the TFH differentiate into full germinal center TFH cells commensurate with the development of the germinal center itself. Those germinal center TFH cells are not restricted to that one germinal center. The germinal center B cells are confined to the germinal center, but the germinal center TFH cells can both exit and re-enter germinal centers and have different phenotypes depending on whether they're within a germinal center or outside of a germinal center. And this results in nomenclature challenges and identification challenges of determining exactly what the history of a given TFH or GCTFH is, because observation of a TFH cell outside of a germinal center, which we generally term a TFH, although some individuals call them a pre-TFH, you can't know in general if that cell has already entered a germinal center and has cycled back out of a germinal center and is on its way back to that same germinal center or a different germinal center, or if that cell has never entered a germinal center. And so that's a nomenclature challenge, which is why we call these cells TFH and these GCTFH, because they clearly have related phenotypes and they can interchange. Subsequent to all of this, TFH cells can go to a resting state and become memory TFH, and that'll be the second section of this lecture. But in terms of T-cell differentiation, those are three key stages of TFH differentiation. And much of TFH cell biology, and certainly differentiation, is closely linked to the microanatomical geography of the T and B cell zones of the lymph node. Early TFH cells migrate to the TB border because they express the chemokine receptor CXCR5, and they downregulate the chemokine receptor CCR7, and that migration is also facilitated by ICOS and PSGL1. CXCR5 is expressed by all B cells and is the main reason B cells localize to the follicle, because the follicle contains the ligand for CXCR5, which is CXCL13. And so it makes sense that TFH cells, to provide help to B cells, have to co-localize with the B cells and express CXCR5 as well. CCR7 is the chemokine receptor important for localization of T cells and other cells in the T cell zone, and so TFH need to downregulate that chemokine receptor so that they can move away from the deep T cell zone that's dendritic cell rich and to the TB border. And so in addition, there's been the very interesting finding that ICOS, which is generally known as a co-stimulatory receptor, also has a role in T-cell migration. And so ICOS signaling can actually induce directional migration towards ICOS ligand-expressing cells. And in mice, all B cells express substantial levels of ICOS, and so ICOS ligand, and so expression of ICOS facilitates TFH entry into the follicle and localization with B cells. PSGL1 binds to the same ligands as CCR7, and so it is thought to anchor cells in the T cell zone, and so TFH downregulate PSGL1 as a way to leave the T cell zone. As the cells further differentiate, the TFH cells actually acquire even higher levels of CXCR5 and will modulate CXCR4, SLAM family receptors, S1P receptors, and perhaps most specifically for germinal center location, TFH cells need to downregulate the molecule, the receptor EBI2. And EBI2 is well characterized to have ligands that are expressed in the follicle, but around the periphery of the follicle and not in germinal centers. And so both germinal center B cells and GCTFH downregulate this receptor, such as to allow them to concentrate in the germinal center zone itself. These stages of differentiation have been well characterized in mice, and this is just example flow cytometry plots. But it's important to point out that the TFH phenotypes are actually well conserved between mice, monkeys, and man in lymphoid tissues of each organism. And certainly CXCR5 is a critical marker in each, but the other migration-associated molecules I mentioned are well conserved in each species, as well are an entire series of TFH phenotypes that define these cells, including high-level expression of BCL6, high-level expression of PD1, ICOS, BTLA, CD200, and quite a few additional markers indicating that, again, overall the TFH differentiation program in TFH biology is very well conserved between species. So TFH differentiation occurs in several stages and involves a number of key migration steps and regulation of migration-associated molecules. Now, what are the signals that control TFH differentiation at each of these stages? What's known is that most TFH differentiation starts at initial dendritic cell priming, and indeed the antigen recognition by the TCR does regulate the likelihood of a cell to differentiate into a TFH versus other CD4 T cell types. And there is an association with TCR affinity, or more accurately, TCR dwell time, between TCR dwell time and TFH differentiation. It is a non-linear relationship, and so there's some association with higher TCR dwell times being associated with TFH differentiation, but in a non-linear way. The earliest non-TCR signal involved in initiation of TFH differentiation that's been identified is IL-6. IL-6 deficient mice have a complete loss of early TFH differentiation, and IL-6 is a molecule that is highly produced by dendritic cells. And we also know that ICOS ligand signaling is important early on for dendritic T cell interactions that drive TFH differentiation. Part of that initial signaling, where the dendritic cell provides TCR signaling, ICOS ligand and IL-6, all of that results in upregulation of BCL6 and CXCR5 and other molecules that are key parts of early TFH differentiation. While it is possible to get CXCR5 expression in the absence of BCL6, and that's certainly been observed for quite some time in human in vitro CD4 T cell studies. You can have at least a transient induction of CXCR5 without additional evidence of TFH differentiation. In the majority of in vivo contexts, certainly in multiple experimental systems, effectively all of the early TFH cells by day 3 in vivo are BCL6, CXCR5 double positive. There aren't CXCR5 single positives, indicating that BCL6 expression and CXCR5 expression are closely linked. Now, if we represent signals involved in TFH differentiation in the classical linear model, or classical sort of rainbow model, where all CD4 T cell differentiation pathways are fully independent and driven by distinct signals, we would diagram it this way, with IL-6 and ICOS being involved, and in the absence of IL-6, IL-21 contributes. But a very important problem in TFH cell biology is that there is no reproducible way to differentiate TFH cells in vitro to date. There have been sporadic successes, but nothing reproducible across many labs, indicating that we still don't know all the signals necessary for TFH differentiation, or possibly that there are inhibitors in the in vitro conditions. And so most of this knowledge is still really dependent on in vivo data. And establishment of a well-defined, well-characterized in vitro TFH condition remains to be done, and would be a major contribution to the field. And we'll come back to mouse-human differences But that summarizes initial TFH differentiation, which can all occur in the complete absence of B cells. Depends on dendritic cells or some other non-B cell APC, but can occur, those first 3 days or so of in vivo differentiation can occur in the absence of B cells. Nevertheless, after that point, TFH cell differentiation becomes extremely B cell dependent. Much of TFH differentiation and function is tightly interconnected with B cells. TFH cells have a highly symbiotic relationship with B cells, and B cells are required for TFH development under almost all in vivo conditions. And what's known about this is that B cells are an abundant source of ICOS ligand, and TFH cells require ICOS and ICOS ligand for their differentiation. And indeed, ICOS ligand deficient humans have serious defects in their ability to mount appropriate immune responses as a result. But so B cells become a source of ICOS ligand, as well as a source of antigen, because as an immune response proceeds, and the B cells receive help from the T cells, those B cells start exponentially dividing, whereas the dendritic cells, at least in an acute, we're talking in the context of an acute antigen exposure, activated dendritic cells will only be around for a few days presenting antigen before they apoptose or die. And so quite rapidly during the course of an immune response, B cells become the predominant class 2 APC available. And so B cells are probably necessary for TFH differentiation because the TFH co-localize with the B cells, right? So they're in the right place at the right time. They selectively provide certain signals to the TFH, ICOS ligand being one of them, probably not the only one, and this needs to be more extensively explored. Certainly CD86 and CD80 contribute, and there are thoughts that other signals from the B cell probably contribute, but these need to be further worked out. And then finally, as I was discussing, B cells really become the primary source of antigen relatively quickly in immune response. And this is key because TFH require TCR signaling for effectively every single round of division in vivo. And so the differentiation and development of these cells is very tightly linked to antigen availability. Another defining aspect of TFH differentiation related to T-cell B-cell interaction is the requirement of SAP, SLAM-associated protein expression, by CD4 T cells for TFH differentiation. And it's been determined that SAP is required for multiple reasons, but one critical reason is that SAP is necessary to facilitate adhesion between TFH cells and germinal center B cells. And in the absence of SAP, which binds to SLAM family receptor cytoplasmic tails and modulates SLAM family receptor adhesion in some way, in the absence of SAP, early TFH differentiation occurs, and the TFH differentiation program looks largely normal, but those TFH cells fail to have adhesion of significant duration to germinal center B cells, and as a result, apparently get signals from the B cell that are of too short a duration to support full germinal center TFH differentiation, and reciprocally then, right, the TFH cells cannot provide sufficient help to the B cells and no germinal center B cells develop. And so again, we see that the TFH cells and B cells have a highly symbiotic relationship and are highly dependent upon each other, because in the absence of SAP, so knocking out the gene SH2D1A, germinal centers fail to develop in virtually any mouse model system that's been reported, whether it be viral infection, bacterial, parasite, protein immunization, or even autoimmune diseases. And indeed, SAP deficiency is a well-characterized human genetic disease, a very rare genetic disease. But these children with the disease called XLP, X-linked lymphoproliferative disease, fail to be able to generate germinal centers and GCTFH and have serious difficulties controlling infections. And in the past, effectively all of those children died by the age of 18. So this all highlights, again, the important role of T-cell B-cell interactions in this process. And as mentioned earlier, the germinal center TFH are not constrained to the germinal center and can both enter and exit this germinal center or other germinal centers, presumably numerous times. And so there's a cyclical aspect of TFH differentiation where presumably a TFH cell can stochastically leave a germinal center, and then due to the lack of continuous antigen recognition on B cells, that GCTFH will become less polarized and become a TFH, which can reside in this follicle or can transit to other follicles. And if that TFH cell then re-engages a germinal center B cell or germinal center B cells, that TFH then, through that high-level antigen recognition by the antigen on the B cells, as well as by the signals, co-stimulatory signals and others provided by the B cells, returns to a full GCTFH polarization. Okay. Now, I've just described a canonical TFH differentiation pathway, which seems to the best of our knowledge, explains most TFH differentiation. But alternative TFH differentiation processes exist, and this is expected. There's no single immutable TFH cell phenotype. It's quite clear in general that CD4 T cells have enormous intrinsic heterogeneity, and TFH cell biology is no different. Some of what we know about this is that while in general Th1 cell responses are desired against viral infections, and Th17 against fungal, and Th2 against bacterial and certain gut infections. Those are either or decisions. In contrast, TFH cell responses are useful against most likely 99% of pathogens, because antibody responses are desired against all pathogens, whether they be viral, bacterial, or fungal. And so therefore, there must be either highly redundant or extraordinarily well-conserved signals to induce TFH differentiation, so that those pathogen recognition signals would occur under almost any possible pathogen infection, right? And so it's most likely, therefore, that multiple different pathogen recognition signals can induce signals within dendritic cells that would induce TFH differentiation. That is to say, there's not going to be a single pathway for induction of those signals because TFH are generated to a wide range of pathogens. IL-6 is most likely a very good example of this. IL-6 is a widely expressed inflammatory cytokine. It can be expressed by dendritic cells, by B cells, but also non-hematopoietic cells. And so that probably builds a heavy layer of redundancy into the process for inducing TFH differentiation. The regulation of ICOS ligand is less well characterized and needs further study. on dendritic cells. It's better understood on B cells. But it's also known that in the absence of IL-6 in mice, there is redundancy with other pathways such that TFH differentiation can still occur, albeit at a delayed or reduced level. And so, for instance, IL-21 or IL-27 can compensate for a loss of IL-6 or limiting IL-6 availability for inducing TFH differentiation. One major complication here comes back to our lack of knowledge of the signals for TFH differentiation to fully recapitulate it in vitro. There is, to date, evidence of apparently, right, major species differences between mouse and humans for TFH differentiation. And this is unexpected because, as I mentioned earlier, phenotypically, whether it be at the protein level or gene expression level, mouse and human and monkey TFH cells, TFH cell biology is very highly conserved. But the available data to date are that in vitro, IL-6 is a very potent inducer of IL-21 in mouse CD4 T cells, and also a moderate inducer of BCL6 in CD4 T cells in vitro. But with human CD4 T cells, IL-6 does not induce IL-21, and nor does it induce BCL6 to any meaningful level. In contrast, in human CD4s, IL-12 is the most potent inducer of IL-21 expression. And while for neither mouse nor humans have strong BCL6 inductive signals been identified in vitro, nevertheless, the current evidence is that the IL-6 is the best identified factor for murine cells. But really, it's a combination of TGF-beta and IL-12 or TGF-beta and IL-23 that are the best inducers of BCL6 in vitro for human cells. And so either there are indeed major species difference between mice and humans for induction of TFH differentiation, or there are aspects to this differentiation that we still don't understand in terms of commonalities between the species. And so this is an area of very active investigation and is quite important because obviously we need to understand the human TFH differentiation signals and signals regulating human TFH much more so than mouse, as it's humans that matter at the end of the day, particularly for biomedical concerns. And so these signals for human TFH differentiation remain incompletely characterized, as do those of mouse cells. And so there's much work to be done in that field. The next topic is TFH cell memory. TFH cell memory was controversial for a while, and a significant amount of that came from an incorrect assumption about the migratory capacities of TFH cells. Some people thought that, like germinal center B cells, germinal center TFH cells could not exit a germinal center once they'd entered the germinal center. And if that were your perspective, it then becomes impossible or difficult to conceive of TFH memory, because germinal center TFH cells would be terminally differentiated cells. That assumption was incorrect. There was early data that has been reinforced in a variety of ways that, as I was discussing in the first section about stages of TFH differentiation, TFH can enter germinal centers, but they can also exit germinal centers and reside in a follicle, and in fact can migrate to other follicles. And TFH cell clones will distribute themselves from follicle to follicle and germinal center to germinal center, unlike germinal center B cells, where a given B cell clone will be restricted to a single germinal center. And so, effectively, as part of TFH differentiation, you can get germinal center TFH cells, but they can leave a germinal center and can then re-enter a germinal center, or can certainly die, or can then go to a resting state and become memory TFH cells. The earliest data on this provided evidence that, just microscopy evidence, that TFH cells could leave a germinal center, and upon leaving the germinal center could change expression of several migration-associated genes, but also upregulate IL-7 receptor. And IL-7 receptor is a classic marker of memory T cells that helps allow for their long-term survival and potentially homeostatic proliferation. And so a simple schematic of TFH differentiation with a focus on memory TFH cells is that TFH can be initially primed in a primary immune response, proceed through to germinal center TFH differentiation, and those cells can interconvert between TFH and GCTFH, and so obviously these are basically indistinguishable. And those cells, again, can either die or go to a resting state and become memory TFH cells that can then, upon a re-exposure to antigen, be reactivated and proliferate and generate new TFH cells. And certainly this polarization is not complete, and some of those cells can be forced to or driven to or happen to also become non-TFH cells upon reactivation. And we will get to this, that there's heterogeneity basically amongst the memory TFH cells as well. It is unclear what induces memory TFH differentiation, if one thinks of it from germinal center T cells. There is certainly evidence that perhaps even TFH cells before a germinal center can actually even perhaps not ever enter a germinal center and develop into memory TFH cells, and this would be supported by data where SAP-deficient CD4 T cells appear to be fully competent for differentiation into memory TFH, even though they cannot fully differentiate into germinal center TFH. It's unclear what signals would drive, as I was saying, conversion from an activated TFH to a memory TFH cell. There may be active signals that do so. I think effectively the null hypothesis would be that as antigen becomes more and more limiting in a germinal center, and perhaps as a germinal center begins to contract, and that, again, antigen becomes limiting, there is less antigen to support a given number of germinal center TFH, and they can either die or leave that antigen-rich environment, and upon exiting that antigen-rich environment have the potential to go to a resting memory state. But that's certainly an area, that's simply a model, and that's an area that requires much more investigation. Okay. So, while it was controversial about whether or not there were TFH, there's very good data both in mouse and man that antigen-specific memory TFH cells exist. those cells can be long-lived. Those cells can be, in the mouse model, adaptively transferred from one animal to another and retain their TFH characteristics and become TFH again upon reactivation and preferentially drive germinal center development after reactivation. Furthermore, memory TFH cells can be present in lymph node and spleen, but memory TFH cells have, in general, the characteristics of central memory CD4 T cells, and like other central memory CD4 T cells, they preferentially reside in lymphoid tissue, but regularly recirculate through the blood. And yes, just like other central memory cells, memory TFH cells can be found in peripheral blood of mice and man. And when looking in peripheral blood, this becomes particularly important in human immunology because the vast majority of the time when doing human immunology, blood is the only type of sample that is available. Lymphoid tissue obviously is much more difficult to come by, and so it's important to glean as much information as possible from blood samples and also understand the limitations of what cells are in blood or not in blood and what characteristics they have. So these are simply example FACS plots of human CD4 T cells. This would be human CD4 T cells, total human CD4 T cells, total antigen experienced CD4 T cells in a lymphoid tissue, tonsil. And in such a tissue, there are a high frequency of germinal center TFH cells shown here that are very high for CXCR5, very high for PD1, very high for ICOS, and very high for both BCL6 and MAF, as well as a population of TFH cells. Now, if you compare that to what's observable in blood, these cells, well, there are CXCR5 positive cells in human peripheral blood, and they are antigen experienced, that is to say they are CD45RO positive or CD45RA negative. However, in contrast to lymphoid tissue, in lymphoid tissue, the vast majority of these GCTFH and TFH cells are activated cells and are actively participating in some antigen-specific immune response. However, in the blood, the vast majority of the CXCR5 positive cells, 99% or more, have a resting phenotype. So therefore, the CXCR5 positive TFH cells in blood are in general very different than the cells identifiable, the bulk of the population identified in tonsil, and that these are by and large activated, currently participating in an immune response, whereas most of the CXCR5 positive cells in human peripheral blood have a resting central memory phenotype. So that is to say they are Ki67 negative, IL-7 receptor positive, CD45RO positive, and have many other markers consistent with central memory type CD4 T cells. And so you can have central memory Th1 cells, central memory Th2 cells, central memory Th17 cells, and central memory TFH cells. Okay. There is a very small population of CXCR5 positive cells in peripheral blood that do have an activated phenotype, and these have been the source of intensive interest in the TFH field, as well as fields of vaccinology and autoimmune disease, because these may represent active TFH cells that are in peripheral blood. This is not a completely resolved field. This topic remains somewhat controversial. But my perspective on these cells, and that of a number of investigators, is that the best available evidence indicates that these cells are related to TFH cells, and that they're related to GCTFH cells, but they are certainly not germinal center TFH cells. They aren't fully differentiated activated TFH cells, that is to say, because they fail to, while they do express CXCR5, PD1, and ICOS, those can be transient activation markers of human CD4 T cells, and definitive TFH markers would be BCL6 and MAF, and say CD200 and several other markers that are preferentially expressed by GCTFH. Those markers are not expressed by this small population of 0.1% to 1% of the CD4s. And so the simplest interpretation of those cells is that there are GCTFH in lymphoid tissue. They can exit and become TFH, and then some of those cells can exit and enter the blood, where you transiently observe those cells in the blood going from an activated state either to a resting state, or going from an activated state to a transient resting state as they go to enter another lymph node and perhaps help facilitate responses in distal lymph nodes. And there's at least one study in mice looking at day 7, early on in immune response, there is an early wave of TFH cells that can be detected in peripheral blood, and presumably the function of that diaspora may indeed be to seed other lymph nodes. Whereas in humans, at day 7, after a flu booster immunization, one can observe a spike in the frequency of those cells, as well as several other conditions. So these cells are frequently called peripheral TFH, so PTFH, or blood TFH. We prefer the longer and somewhat less appealing, but I think more accurate phrase for the moment, of activated TFH-like cells. Whereas in contrast, the vast majority of the CXCR5 positive population, again, are resting central memory TFH cells. Now, if you take that large population of memory TFH cells, one can observe that there is extensive heterogeneity amongst those cells. This is quite normal for CD4 T cells and even CD8 T cells. I mean, it's been observed with high multi-parameter analysis via CyTOF type mass spec flow cytometry, that antigen-specific memory CD8 T cells can actually have effectively hundreds of different distinguishable phenotypes, and that there's not a specific central memory CD8 T cell phenotype, but instead effectively a cloud of related phenotypes. And CD4 T cells are even more heterogeneous than CD8 T cells, and so it's not surprising that when looking at memory CD4 T cells, as one explores more and more phenotypes, that is, different cell surface markers or intracellular markers, one can observe greater and greater heterogeneity, which perhaps is a means for the immune system to avoid pathogen escape by not having a singular focused differentiation program, instead a differentiation program that nevertheless samples a variety of related and alternative outcomes. So among that heterogeneity, some populations are more prominent in terms of their frequency compared to others. And one such marker is PD1. And PD1 is of interest, of course, because it is very highly expressed by TFH cells. TFH cells, germinal center TFH cells, are perhaps the highest expressing PD1 positive cells in the body. And so PD1 is part of the TFH differentiation program. it is regulated in some way by BCL6. It is also a TCR activated gene, and so it can be either a marker of activation or TFH differentiation or both. But there is a popular, and so these PD-1 high cells are in fact activated proliferating cells. But these PD-1 low cells, it's been observed, are resting memory cells. And indeed, by gene expression profile, they tend to have the most TFH biased gene expression profile compared to the PD-1 negative CXCR5 positive cells. And memory TFH cells can be stratified by additional markers, and these cells seem to have or do have distinguishable functions. And we observed, for example, that the PD-1 low, the PD-1 expressing memory TFH cells in humans that do not have a Th1 bias, that are CXCR3 negative, were the cells that had the greatest functionality for B-cell help in vitro. And one can certainly observe memory TFH cells that do have some Th1 bias or Th2 bias or Th17 bias in peripheral blood, and that will surely modify the function of those cells as well. And we'll come back to that topic later on when discussing complexities of CD4 T cells, of TFH cells. Sorry. And so it's unclear, it's not known where in lymphoid tissue memory TFH cells reside, but there's at least some evidence that they preferentially reside, and it would be logical that they would preferentially reside near the TB border as opposed to deep in the T cell zone, since they do express CXCR5 as well as CCR7. And interestingly, memory TFH cells have low levels of BCL6, that is to say, their expression of BCL6 is indistinguishable from other resting memory cells. This is not particularly surprising for central memory cells, because central memory cells generally do have a very quiescent phenotype and reacquire many of their lineage-defining characteristics upon reactivation. And indeed, recently it's been observed that when memory TFH cells get re-stimulated by B cells, that rapidly induces re-expression of BCL6, specifically by the memory TFH cells. The next topic is TFH transcriptional regulation. CD4 T cell differentiation is generally characterized by a distinct canonical transcription factor associated with each different outcome, and BCL6 is that transcription factor for TFH cells, frequently referred to as a master regulator transcription factor. However, it's abundantly clear that more than one transcription factor is necessary for any of these different CD4 T cell differentiation pathways. And as such, transcription factors such as BCL6 or TBET or GATA3 are generally now referred to as lineage-defining transcription factors, because they are critical for each of those distinct differentiation pathways, and are also very useful for distinguishing different types and functions of CD4 T cells. Any form of transcriptional regulation in mammalian cells involves numerous transcription factors acting in coordination, but there are good data now indicating that any particular cell type can likely be distinguished from another by expression of perhaps 4 to 6 different transcription factors in different levels. For TFH cells, there are a number of transcription factors that have been shown to be important for TFH differentiation and/or function. Most prominently of these, and most centrally, is BCL6. But in addition to that, there are these other transcription factors. And one way to think about this is that there are transcription factors that have more broad function in CD4 T cells and are activated downstream of initial TCR activation and co-stimulation, which can activate or drive the expression or repression of expression of a broader network of genes, and then expression of lineage-defining transcription factors can hone that those initial signals to refine differentiation towards a particular TFH T cell differentiation program. So, for example, IRF4 and BADF are necessary for TFH differentiation. BADF is an AP1 family member component, and AP1 proteins in general are essential for T cell activation and proliferation. And BADF and IRF4 both have roles in CD4 T cell differentiation pathways other than TFH as well. For example, Th17 differentiation. MAF is critical for TFH differentiation, but can also have roles in other CD4 T cell pathways and functions. And similar things can be said about STAT proteins. For STATs, the most important STAT in mouse CD4 T cell differentiation to TFH is STAT3, and STAT3 is a critical regulator of IL-21 expression among numerous other protein or genes of interest. STAT3, however, of course, has roles in other types of CD4 T cell differentiation and is not completely restricted to TFH cells. In the absence of STAT3, or perhaps low levels of STAT3, STAT1 can actually compensate for STAT3 function in murine TFH differentiation. And redundancies such as these are probably essential for survival, so that the immune system can recognize and respond to a wide variety of pathogens and conditions, so that escape cannot occur. Now, while STAT3 and STAT1 are important for murine CD4 T cell TFH differentiation, and STAT4 has some role, in humans it appears that either STAT3 and STAT4 have equivalent roles, or that STAT4 may even have a more prominent role. This is based on both in vitro data, where for mouse CD4 T cells, STAT3 is essential for IL-21 expression, but for human CD4 T cells, it's STAT4 that is essential. And in addition, mutations in STAT3 and STAT4 in humans can both result in reductions in memory TFH frequencies in peripheral blood, indicating that both molecules do have roles. BADF and IRF4, as I mentioned before, are both early transcription factors that are certainly important for TFH differentiation, but also related or other CD4 T cell programs. E proteins, which are critical for a wide variety of processes in B cells and T cells, are also, perhaps not surprisingly, important for TFH differentiation. In particular, the E protein ASCL2 has been noted to be important for CXCR5 expression, but other E proteins can function in a related function, in a related manner. MAF is highly expressed in TFH cells, and really, in terms of markers, if one looks for protein expression, it's really BCL6 expression and MAF expression that most distinguish TFH cells from other types of CD4 T cells. And MAF is an important regulator of IL-21 and IL-4 expression, as well as CXCR5 and other molecules. Furthermore, it's not only conventional transcription factors, but it was recently identified that, interestingly, the transcription elongation factor, cyclin T1, preferentially regulates TFH versus other CD4 T-cell differentiation programs, indicating that regulation of epigenetics and transcription elongation are probably also central players in these cell fate decisions. While those are transcription factors that are important for induction of TFH differentiation, there are also molecules, transcription factors, that inhibit TFH differentiation. The first of these that was identified is BLIMP1, and BLIMP1 is a powerful antagonist of TFH differentiation. BLIMP1, both in B cells and in T cells, potently represses BCL6 directly, and so whereas BCL6 potently represses BLIMP1 directly, and so there's a potent reciprocal antagonism between these two transcription factors, BCL6 and BLIMP1, regulating TFH versus non-TFH differentiation. STAT5 is also known to inhibit TFH differentiation. In part, this is because IL-2 signaling, which is the cytokine that is the strongest inducer of BLIMP1, IL-2 signaling induces BLIMP1 via STAT5, and so STAT5 induces BLIMP1. But in addition, STAT5 inhibits other aspects of TFH differentiation as well. A third category are, in fact, Forkhead box transcription factors, such as FOXO1 and FOXP1. These Forkhead box proteins are well known to regulate T-cell quiescence and also a variety of aspects of T-cell migration. And it's been observed by multiple groups that Forkhead box proteins can potently inhibit TFH differentiation specifically, and that it's necessary to repress those molecules to actually allow TFH differentiation to occur. It's also been shown that TBET inhibits TFH differentiation, and one mechanism by which it does so is to actually directly bind to BCL6 and limit BCL6 functionality. BCL6, again, is the lineage-defining transcription factor for TFH cells. How BCL6 regulates TFH differentiation is still incompletely characterized, but many things are being learned, and that knowledge is accelerating. BCL6, at least in B cells, is known to be exclusively a repressor, and so it's not clear whether BCL6 turns on any genes directly in TFH or not. That's still to be determined. But generally speaking, the regulation of TFH, gene regulation, can be characterized in the following schematic manner, where one can conceive of 4 different major categories of TFH-associated genes: products of TFH cells, either secreted molecules or adhesion molecules that are important for helping B cells; localization of the TFH cells; differentiation of the TFH cells; and repression of alternative fates. BCL6 contributes to regulation of genes in each of these categories, but not all genes, certainly, in these categories. And BCL6 has multiple functional domains, and in fact, frequently BCL6 is thought about, I think, in an oversimplified manner, and this is a more realistic schematic of how BCL6 may regulate TFH differentiation. BCL6, shown in blue here, is known to interact with a wide range of proteins. And the BTB domain is a well-characterized protein-protein interaction domain. It actually is necessary for BCL6 dimerization with itself, which isn't shown here, but also BCL6 interaction with other co-repressors, as well as direct binding to histone deacetylases. Itch is a ubiquitin ligase that can, in fact, monoubiquitinate BCL6, and it binds in this long middle domain of BCL6, which also contains pest degradation sequences, as well as a very interesting acetylation motif that has been best characterized in germinal center B cells, where BCL6 can be acetylated, and that acetylation actually prevents interaction of BCL6 and MTA3, which is a co-repressor that can recruit histone deacetylases in a cell type specific manner. The zinc finger domain is frequently simplified as the DNA binding domain, but in fact there's 6 zinc fingers, and only 2 of which are necessary for DNA binding, and the other 4 zinc fingers, as well as other regions of the zinc finger domain, are probably central protein-protein interaction domains for BCL6 to interact with other transcription factors and chromatin modifying proteins. And as such, between BCL6's primary sequence shown here and the different categories of genes that get regulated in TFH differentiation, it appears that BCL6's functions in TFH cells are tightly interwoven into a variety of different components of TFH differentiation and biology. The next topic is the concept of layers of differentiation. A simple, conventional, what I would call rainbow view of CD4 T cell differentiation is shown here, where TFH, TH1, TH2, TH17, and Treg are all sort of fully independent CD4 T cell types and fully independent T cell differentiation programs. That isn't accurate. There's no single visualization that can accurately capture CD4 T cell heterogeneity in biology. This is certainly a useful schematic, but one area in which it falls short is in considering aspects of class switch recombination, for instance. TFH cells produce IL-4 and IL-21, which are important for class switch recombination from IgM to IgG, but there are other class switch recombination events that can be influenced by Th17, Th2, or Th1 type cytokines, and the question is, how do those cytokines get produced by TFH cells? Those cytokines can be produced by TFH cells, at least at low levels, and the model that I would put forward for thinking about this process is shown here, where one can visualize a cell, the inside of a cell, in the way that one would visualize an architectural diagram, a blueprint diagram of a building with different floors, each floor with a different blueprint. And the TFH program, as just described, consists of several modules, products of TFH location and differentiation, and those genes are controlled by BCL6 and other TFH-associated transcription factors. But a TFH cell can also still have some activity of a Th1 gene program or a Th17, or not shown here, a Th2 gene program, such that BCL6 is continuously repressing any attempt by a cell to express a Th17 or a Th1 program. And one can observe this by looking in human TFH cells, where there's really a very low percentage of them that will make IL-17 or interferon gamma, for instance. But there can be some production of those cytokines, and certainly some evidence for low-level activity of Th1 or Th17 or Th2 programs in TFH cells. And those can be driven certainly if there are continuous external signals coming into the cell that are inductive signals, for example, for a Th1 program, BCL6 is going to attempt to repress those, but cannot completely extinguish them. And so you can get some level of coexistence between TFH and other programs within the same cell. There are many complexities to TFH differentiation in biology and many unresolved aspects of TFH, even though really enormous progress has been made in the past 5 years. And one is that a conventional model of TFH differentiation, or of CD4 T cell differentiation in general, like this, where TFH are separate from Th1, 2, and 17, is the primary model, and I think generally accurate, and that should be considered in combination with this layers of differentiation model that I just summarized. There are alternative models, and one which I'll discuss right now. One alternative model would be that, or is that, TFH is effectively sort of a secondary program, and that an immune response first generates Th1, 2, or 17, and then those cells can subsequently additionally turn on a follicular helper type program. And a variant of that idea would be this, where in a given immune response, really everything would be a Th17 cell, for instance, and that you would have Th17 effector cells as well as Th17 FH cells, follicular helper cells. That model has strengths, and one of the strengths comes from the observation that you can find TFH cells that will express interferon gamma or IL-13, potentially, or IL-17, particularly under extreme conditions. And so the observation that a TFH cell is not necessarily exclusively a TFH and can have some aspects of Th1, 2, or 17 is consistent with this alternative model. However, I think this type of model is more accurate and more useful, because while it's appealing to have a simple shared nomenclature, that is inherently problematic for cells as heterogeneous as CD4 T cells. And really, from a semantic perspective, this is in part a question of the perceived primary biology of the cells in question. For example, is a given cell primarily a TFH cell helping B cells, or is it primarily a Th1 cell inducing inflammation in an infected tissue? And I think Tregs are really an illustrative example here. While they're clearly T-bet positive or ROR gamma T positive Tregs, nevertheless, those cells are still Tregs, as their primary biology is to dampen or regulate immune responses. And so while you can have certainly complexity to Tregs, nevertheless they are Tregs based on their primary function. That same conceptual model holds for TFH cells. The job of a TFH cell, the function of a TFH cell, is primarily to help B cells, and therefore that really is the primary program as well as function of those cells, even though one can certainly find TFH cells that express some attributes of Th1 or Th2 or Th17 cells and T-bet. Furthermore, as discussed earlier, TFH differentiation can be observed to be occurring, to be polarizing from as early as the second cell division in vivo, and therefore there's good evidence from very early time points during dendritic cell priming that the dendritic cell and the inflammatory environment are inducing a given T cell to either differentiate towards a TFH pathway or a Th1, Th2, or Th17 pathway. Within the category of conceptualizing different CD4 T cell types and differentiation pathways, follicular regulatory cells, that is TFR cells, are a different topic, and that's because TFR cells have been identified as a subset of natural Tregs, so developed in the thymus before maturation of naive mature T cells, where effectively all natural Tregs develop in the thymus, and a subset of those can express BCL6 and become TFR cells, which are Tregs that also have an ability to express BCL6 and CXCR5, and therefore will migrate to and localize in follicles and restrain B cell responses. TFH functions. TFH are responsible for T cell help to B cells, and the most prominent role of TFH cells is their necessity for germinal centers. The germinal center is the primary site of affinity maturation of B cells, as well as important for extensive proliferation of B cells and the production of memory B cells and long-lived plasma cells. But really, the extraordinary process of somatic hypermutation and selection, the affinity maturation, is one of the miracles of immunology. A germinal center is, in essence, evolution in miniature, as the immune system attempts to keep up with a pathogen in the course of a single infection, and to develop truly high affinity B cell responses to that pathogen. And TFH cells are necessary for the initiation of the germinal center, the function of the germinal center, or functions of the germinal center, which would include the B cell survival, proliferation, hypermutation, and affinity maturation or selection of the cells, as well as the TFH are also required for the products of the germinal center, the memory B cells and long-lived plasma cells. And so, therefore, what functions do TFH cells have, in germinal centers that are so critical? Globally, the experimental data is that T cell help, TFH cell help to the B cells is really the critical limiting factor for germinal center size and germinal center function, where the function of a germinal center occurs by antigen retention by FDCs. B cells compete to bind that antigen, and then the B cells internalize that antigen, process it into peptides that can be presented on class II molecules, and then the different B cells present those peptides back to TFH cells, and the TFH cells conceptually recognize the different amounts of peptide on the surface of the different B cells as a proxy for the affinity of each given B cell, whereby germinal center B cells that have a mutated BCR with a higher affinity for the antigen will, as a result, as a consequence, present more peptides on their surface because they will have acquired and processed more antigen. And therefore, those B cells will obtain more help from the TFH cells, and on a competitive basis will become the primary clone in the germinal center, and that process then iterates over and over. And strikingly, the amount of proliferation of a given B cell appears to be directly correlated to the amount of T cell help that germinal center B cell receives. So, what are the molecular mechanisms of TFH help to germinal center B cells? First of all, part of it is indeed localization. TFH cells provide help to B cells in part because they co-localize with the B cells, and the TFH are the cells that are physically in the germinal center, so those are the cells that provide help. However, migration alone is not sufficient to explain the functionality of TFH cells. Being in the right place at the right time is part of their biology. But TFH cells also provide specific signals to B cells, and we'll focus on germinal center B cells, that play roles in the survival, proliferation, and plasma cell differentiation, hypermutation, and class switch recombination, as well as attraction and adhesion. And so survival signals are critical because germinal center B cells are highly proapoptotic. A germinal center B cell left alone will generally die by apoptosis within approximately 6 hours. And so they continuously require extrinsic signals for their survival. And those signals, some of those signals may come from FDCs or stroma, but definitely major signals, major necessary signals come directly from the TFH cell. CD40 ligand and IL-4 are probably the two most prominent survival signals, although it's also been reported that TFH can produce BAFF, and BAFF is a major survival factor for B cells as well. Then B cells that have presented antigen to the TFH cells and receive those survival signals also need to proliferate, to expand in the dark zone. Selection occurs in the light zone, expansion occurs in the dark zone. Light zone effectively is the TFH-rich region next to the FDCs. The dark zone is the opposing, the opposite region of the germinal center, which is has few TFH, but is enriched for proliferating germinal center B cells, which will then cycle back up to the light zone for additional selection. So what are the TFH cell signals that are involved in proliferation? There's not a single signal, just like survival. It's probably quite important that each of these processes be controlled by a multifactorial signal from, multi-component signal, I should say, from the TFH cells, so as to best regulate the process. Certainly CD40 ligand expression by the TFH cells is essential. If one blocks CD40 ligand, germinal centers collapse almost immediately. And IL-21 is also a very potent proliferation signal. However, IL-21 is really the most potent signal known for inducing B cell differentiation into plasma cells. And so while IL-21 provides not exclusively plasma cell differentiation signals, it is the most potent cytokine known for inducing plasma cell differentiation. IL-21 and IL-4 together both contribute to, are both TFH products that help induce AID and maximize BCL6 expression, both of which are necessary for hypermutation and survival of hypermutation by germinal center B cells. There are almost certainly still unknown factors being produced by TFH cells that are important actually for each of these processes, and so these remain active areas of exploration. But this is our current understanding of them. Class switch recombination, I discussed some earlier. Really, IL-21 and IL-4 together are potent inducers of class switch recombination from IgM to IgG, and in humans, IgG1 is the dominant IgG isotype and is also broadly functional against a range of pathogens, and is the dominant class switch induced by the presence of high levels of IL-21 and modest levels of IL-4, which is what TFH appear to generally provide. Whereas if very low levels of IL-21 are around and very high levels of IL-4 are around, IgE class switch recombination occurs, and this can be observed, for example, in patients that have deficiencies in IL-21 production. Additional functions of TFH in terms of their help to B cell are molecules involved in attraction and adhesion. The germinal center is a very tightly confined, for the B cells, microanatomical niche, and the TFH cells actually continuously secrete CXCL13, most likely to attempt to retain germinal center B cells in that location. And the TFH cells also express a range of adhesion molecules that are critical for their physical interaction with the B cells and regulate almost certainly the duration and the quality of help provided by the TFH to the B cell. For example, the stronger that adhesion, the longer the duration of contact, and therefore the more extensive the signals, quantitatively and qualitatively, can be transferred from the T cell to the B cell. For example, the amounts of IL-21, as well as probably other quantal processes. And the magnitude of that help drives the extent of B cell, germinal center B cell proliferation in the dark zone, and therefore there's a direct relationship between adhesion and the T cell help. It's more complicated than that because there are adhesion molecules that regulate the interaction in both positive and negative ways. SLAM-F6 is a critical example of this, that can either enhance or inhibit adhesion or the duration of adhesion between a TFH cell and a B cell, depending on accessibility of SAP. And the signals that I just described are effectively all positive signals, except for SLAM-F6. Negative signals are also a major component of inhibitory receptors, I should say, are a major component of TFH cell biology. Not only very high level expression of PD-1, but also very high level expression of CTLA, of BTLA and CD200, as well as additional inhibitory receptors. These inhibitory receptors probably do more than one thing to the TFH cells, but one basic aspect of their functionality is to inhibit TFH proliferation in response to antigen. TFH cells are constantly seeing antigen being presented by B cells, but the role of a germinal center is for the B cells to proliferate, because it is the B cells that are mutating and evolving, not the TFH cells. And therefore, for a given germinal center of a given size, a stable number of TFH cells is needed. And therefore, once a germinal center has formed, the TFH cell numbers should stay relatively stable, and the TFH should, generally speaking, avoid proliferating and thereby avoid taking over, essentially, the germinal center. And therefore, for a T cell to see large amounts of antigen and not proliferate and not apoptose most likely involves interplay of these inhibitory receptors on the surface of the TFH cell. TFR cells, that is, T follicular regulatory cells, also have roles in germinal centers, and they have an inhibitory role. And again, these cells are natural Tregs that express BCL6 and CXCR5 and therefore have the capacity to co-localize with TFH cells and germinal center B cells, and thereby provide Treg type functions in that environment, similar to how TBEP positive Tregs will co-localize with Th1 cells and thereby limit their activity. The functions of TFR cells remain to be fully understood. To date, antigen-specific TFR cells have not been described, that is, to regulate in an antigen-specific manner, and it remains unclear whether they act directly on primarily act directly on the TFH cells or the germinal center B cells or both. The best available data indicate that their function is to prevent the generation of autoreactive B cells in germinal centers as those B cells are rapidly mutating. TFH cells in diseases. TFH cells are essential for the generation of most isotype-switched antibodies and affinity maturation, and therefore they have an obvious role in protective immunity against infectious diseases. Antibodies are necessary for control of chronic LCMV infections in a mouse model, and defects in TFH cell frequencies have dramatic effects in failure to control LCMV, for example. Interestingly, LCMV has a surface that's very difficult for antibodies to bind to, and therefore, conceptually, it may be that you need particularly good T cell up to B cells when the target of the B cells, the target of the antibodies, is a particularly difficult target for which to develop high affinity B cells. One can imagine that the greater the mutational landscape that the B cells have to explore, the greater the T cell help that's necessary. Or one can imagine that the lower the affinity of the initial naive B cells, the greater the T cell help may need to be to keep those B cells alive and proliferating. A similar problem exists for HIV, where it's very difficult. It appears to be very difficult to find B cells that can bind with high affinity to the key neutralizing antibody targets on HIV that are conserved, and this problem is made even worse in the context of HIV by the extreme mutability of that virus. And it turns out that TFH frequencies are in fact associated with the amount and the quality of antibody responses against SIV, the non-human primate version of HIV. And indeed, impaired TFH help to B cells is observed in HIV-infected individuals, which appears to exacerbate the difficulty in generating neutralizing antibodies against HIV. Conversely, interesting, there are rare individuals who make very potent neutralizing antibodies against HIV, and these antibodies are referred to as broadly neutralizing antibodies. One important question has been, why are there rare individuals who can make these antibodies, but most HIV-infected people cannot? And interestingly, it turns out that those individuals who do make these high-quality, broadly neutralizing antibodies against HIV, those individuals have unusually elevated frequencies of highly functional memory TFH cells, these PD-1 positive, CXCR3 negative memory TFH cells. People with high frequencies of those highly functional memory TFH cells are more likely to make broadly neutralizing antibodies against HIV than other individuals. This all again is consistent with the hypothesis that TFH are a key limiting resource for the development of high affinity B cell responses. This is essential for people who think about vaccines, because almost all licensed human vaccines work on the basis of long-term protective antibody responses, and therefore being able to enhance or increase TFH cell frequencies may be an excellent approach for better vaccine design. Indeed, even immunotherapeutic approaches are conceivable there, because there was a dramatic result in a mouse malaria model whereby providing antibodies that increased the TFH response or TFH functionality resulted in rapid control and clearance of mouse malaria. In addition to protective immunity against infectious diseases, TFH have roles in regulation of intestinal microbiota, and this appears to be through similar mechanisms where The intestinal microbiota consists of commensals and some pathogens, and TFH cells are important for maintaining the B cell and antibody responses in the intestine, and appear to be important for the majority of the IgA that gets produced, that controls the intestinal microbiota. And this can have complex implications, in addition to simple control of the abundance of bacteria in the gut. While there's lots of interest in TFH cells in the context of protective immunity to pathogens, there's at least as much interest in TFH cells, and at least as important of a role of TFH cells in autoimmune diseases. TFH cells are important players in many mouse models of autoimmune diseases, as well as human autoimmune diseases, and several different categories of such autoimmune diseases. TFH are associated with autoimmune diseases that have autoantibodies, some diseases that have autoantibodies as a pathogenic component, but also autoimmune diseases that have autoantibodies as a biomarker, or just a component of the response. In addition, there are associations between TFH and autoimmune diseases that don't appear to have a major antibody component at all. That appears to be true for autoimmune diseases like systemic lupus, where autoantibodies are considered primarily markers of disease, not causes of disease. But interestingly, in humans, a common variant of SLE, lupus, is lupus nephritis, which can result in severe kidney dysfunction associated with inflammation and accumulation of high concentrations of antibodies. But importantly, actually, it's been found that there are clusters of TFH cells and B cells in the kidneys themselves in those infected individuals, indicating that the TFH are actually somehow directly causing or exacerbating the autoimmune disease in that kidney tissue itself. And so therefore, these and other data suggest that TFH may be major regulators of ectopic follicles in autoimmune diseases in general. And as such, TFH may contribute to autoimmune diseases both by facilitating antibody responses and by facilitating and maintaining lymphoid structures that may, such as ectopic follicles, which serve as nucleation points for other cells that may be directly pathogenic for that particular disease. TFH most likely have important roles for allergies, but this is an area that actually needs further investigation. There was one well-designed mouse study that showed that TFH were essential for IgE responses. That was in the context of a helminth infection, but that is likely to also hold true for IgE production in allergic diseases. But again, that remains to be well established in different types of allergies, both animal models and in humans. Most likely, the most surprising finding in the past couple of years in terms of diseases that TFH are present in is cancer immunity. There were two independent studies in the past year that found that TFH cells are actually a strong positive biomarker for long-term survival of human cancer patients in two very different types of cancer. One was a colorectal cancer study, and another was a breast cancer study. And in each study, the groups were not looking for TFH cells. They were, generally speaking, looking for most likely CD8 T cell correlate of not protection, but long-term survival of people with these cancers, and were doing gene expression profiling, and found actually a TFH signature was the most prominent signature in each of those different cancers. And so it's unclear what role TFH may be playing there, but one possibility is that they're helping serve to nucleate other cells that may have a more direct protective role, but this remains to be seen. Overall, clearly TFH cells have important and varied and multifaceted roles in diseases, and while there's been a great deal of activity and progress in studying TFH cell biology over the past 5 years, there is a great deal of work still to be done, and hopefully will lead to utility of TFH cells, manipulating TFH cells in the context of enhancing protective immunity to infectious diseases, in the context of vaccines, as well as in manipulation of TFH cells in other contexts, perhaps in allergies, but certainly in autoimmune diseases with strong B cell and autoantibody components, where blocking TFH cells, since TFH cells can be limiting factors, T cell up to B cells can be limiting factors for many B cell and antibody responses. Blocking TFH cells may have effective therapeutic effects in particular diseases. Thank you for your time.